It's already happening. Right now, deep inside the structure of your brain in regions you've never consciously felt, dopamine is moving through neurons the way it always has, but the environment around those neurons has shifted. Testosterone, which once moved through your bloodstream in higher concentrations, has been declining. Not suddenly, not dramatically, just steadily, across months and years, dropping in ways too gradual to notice in any single moment. Welcome to the sleepy biologist. This is a place for rest, for curiosity without urgency, for letting biology unfold in the background while you settle into stillness. If you're already lying down, already drifting, you're exactly where you need to be. I'm grateful you're here, and I'm especially grateful to the members of this channel who make these long, quiet explorations possible. Your support through membership, through super thanks, through simply being here, it matters. If you'd like to join that community, the link is always available. If you'd like to leave a comment about where you're listening tonight, I always love hearing from you. And if a like or subscribe feels right, it helps others find this space too. But for now, let's just be here. Let's think about dopamine. Let's think about what happens when the molecules that once shaped how your brain experienced reward begin to change their presence. You start to notice it in small ways first. Maybe a song you love doesn't hit the same way. Maybe a hobby that used to pull you in now feels optional. Maybe accomplishment still registers, but the surge of satisfaction feels quieter, less sharp. You might assume you're tired. You might assume it's age or stress or circumstance, and maybe it is, but it's also chemistry. It's also the slow recalibration of neurons that have been listening to testosterone for decades. Testosterone doesn't make dopamine. That's not how it works. But testosterone influences the neurons that produce dopamine, the receptors that receive it, and the pathways that carry its signals through your brain. It acts like a background amplifier, adjusting the volume on certain experiences. When testosterone levels are higher, dopamine-related processes tend to operate with more intensity. When testosterone begins to fall, the amplification weakens, the system doesn't break, it adjusts. The neurons themselves are still intact, they're still firing. They're still releasing dopamine into the synapse, that tiny gap between one cell and the next. But the receptors on the receiving neuron, the proteins that catch dopamine molecules and translate them into electrical signals, are starting to change. Some are being removed from the cell surface. Some are becoming less sensitive. The cell is recalibrating based on what it's been receiving lately, and what it's been receiving is less androgenic input. This happens in a region called the Ventral Tegmental area, a small cluster of dopamine-producing neurons buried deep in the midbrain. You've never felt it directly, you never will, but you felt its effects your entire life. Every time something felt rewarding, every time motivation surged, every time you wanted something badly enough to move toward it, this region was involved. And now, with less testosterone binding to androgen receptors scattered throughout this tissue, the baseline activity of these neurons is shifting downward. It's not a collapse, it's not depression, not inherently, it's recalibration. The brain is incredibly adaptive, and it doesn't interpret falling testosterone as a crisis. It interprets it as new information, and it adjusts accordingly. You might find yourself less driven by competition, less interested in proving things, less pulled toward risk. These aren't failures of character, they're neurochemical shifts. Testosterone had been biasing your dopamine system towards certain kinds of goals, assertive ones, physical ones, status-related ones. Without as much of that bias, your dopamine system starts responding more evenly to different types of reward. Quieter rewards, social connection, stability, reflection. Some people experience this as loss, Some experience it as relief. Most experience it as both, depending on the day. There's a part of your brain called the Nucleus Accumbens, a small structure involved in translating dopamine signals into the feeling of wanting. For years, testosterone helped amplify the signals arriving here. When you achieved something, when you anticipated something pleasurable, the dopamine surge in the Nucleus Accumbens was stronger. The feeling of reward was sharper. Now, with less testosterone modulating the system, the surges are smaller, the peaks are lower. The experience of reward becomes less about intensity and more about presence. You still feel pleasure, you still feel satisfaction, but it arrives differently. It lingers differently. It doesn't announce itself the way it used to. And here's something interesting. Your brain isn't passive in this process. It's not just accepting lower dopamine signaling and moving on, it's adjusting the density of dopamine receptors. In some areas, it's growing more receptors to compensate for weaker signals. In other areas, it's removing receptors because they're no longer being stimulated as often. This is called receptor regulation, and it happens constantly, invisibly, in response to the chemical environment each neuron experiences. So even though testosterone is lower, even though the dopamine system is operating at a different baseline, the brain is working to maintain some version of equilibrium. It's trying to keep you functional. It's trying to keep reward meaningful, just has to do it with different tools now. The prefrontal cortex, the part of your brain responsible for focus and goal-directed behavior, also relies on dopamine. And it also relied on testosterone to modulate that dopamine activity. With less testosterone, the prefrontal cortex doesn't lose its ability to focus, but it does become more selective. It takes more effort to sustain attention on things that don't feel immediately relevant. Motivation becomes harder to summon on command. You can still do the work, you can still think clearly, but the internal sense of urgency is quieter. And maybe that's not entirely a bad thing. Maybe urgency was exhausting. Maybe the constant pull toward achievement, toward competition, toward proving yourself, maybe that was never sustainable. Maybe this quieter version of motivation is actually closer to what your body needs now. Your dopamine system is still working. It's still capable of joy, of interest, of desire. It's just working within a different hormonal context. And over time, as your brain continues to adapt, you might find that satisfaction comes from different places. Smaller places, steadier places. The shift is gradual, the shift is ongoing, and the shift is already inside you, unfolding in the silence between thoughts, in the spaces where neurons meet and molecules travel, and your sense of what matters slowly, quietly begins to change. What stays when urgency leaves? What remains when the edge softens? Somewhere in the midbrain, in the space between signal and response, the answer is forming. Not in words, just in chemistry, just in time. You've probably felt this before. The sense that your body remembers things your mind has forgotten. A neuron doesn't think, it doesn't plan, but it does respond to patterns. And after years of responding the same way to the same chemical signals, it develops something like memory. Not the kind you access consciously, the kind that lives in structure, in the shape of proteins, in the number of receptors embedded in a cell membrane. These dopamine neurons in your midbrain have been listening to testosterone for as long as you've been alive, not directly. Testosterone doesn't bind to dopamine receptors, but it binds to androgen receptors inside and around these cells. And when it does, it influences which genes get turned on, which proteins get built, how much dopamine the neuron can produce, how sensitive it becomes to incoming signals. For decades, this process has been happening at a certain level, a certain intensity, and now that intensity is dropping. The neurons notice. They notice because the androgen receptors they contain, those proteins designed to catch testosterone molecules and relay instructions into the cell's nucleus, are receiving fewer activations. The receptors are still there, still functional, just less occupied. And when a receptor sits empty for long enough, the cell starts to adjust. It's a slow adjustment. Weeks, months, maybe years, but it's measurable. Researchers can see it in animal models. They can see it in tissue samples. When testosterone drops, the expression of certain genes inside dopamine neurons changes. Genes that code for enzymes involved in dopamine synthesis. Genes that regulate how much dopamine gets packaged into vesicles, those tiny bubbles that store neurotransmitter molecules until they're ready to be released. The cell doesn't stop making dopamine, it just makes less per cycle, and it releases less per signal. You already know this feeling, even if you've never named it. The feeling of wanting something but not quite enough to move toward it. The feeling of enjoying something but not enough to seek it out again. It's not absence, it's attenuation. The signal is still there, just quieter. There's a specific type of dopamine neuron that projects from the ventral tegmental area to the nucleus accumbens. This pathway is central to motivation, to desire, to the feeling that something out there is worth pursuing. These neurons have androgen receptors. Not every dopamine neuron does, but these ones do, and when testosterone levels fall, these particular neurons begin to change their firing rate. They become less responsive to stimuli that used to excite them. The burst of dopamine they release when you encounter something rewarding, it's smaller now. The pause in firing when a reward is withheld, it's less pronounced. The neuron is still communicating, it's still part of the network, but its voice is softer. And the neurons downstream, the ones receiving that dopamine signal, start to adapt too. They adjust their receptor density. If dopamine arrives less often, or in smaller amounts, the receiving neuron might upregulate its receptors. Grow more of them. Try to catch every molecule it can, or it might downregulate. Remove receptors. Accept the new normal. Which direction the adjustment goes depends on a lot of things: age, genetics, how quickly testosterone dropped, whether other compensatory systems are kicking in. But either way, the system is changing. It's interesting, isn't it? How something you've never consciously controlled has been shaping your sense of what matters. How a molecule you didn't choose to produce has been influencing which goals felt urgent and which felt optional. And now, as that molecule's presence fades, the neurons that relied on it are rewriting their own instructions. Not dramatically, not all at once, just steadily. In the Substantia Nigra, another cluster of dopamine neurons, the same thing is happening. These neurons are more involved in movement, in the smooth execution of physical actions, but they also have androgen receptors, and they also respond to testosterone's decline. The change here is subtler. You might not notice it as motivation. You might notice it as a slight hesitation before standing, a fractional delay in initiating movement. Nothing you'd call a symptom, just a texture, a barely perceptible shift in how your body responds to the intention to move. The dopamine neurons are still firing, but the baseline rate is lower. The peak rate during movement is less sharp. And over time, the motor cortex, the part of your brain that plans and executes movement, starts to notice that the dopamine feedback it's receiving is different, not absent, different, so it recalibrates too. There's a part of this process that feels almost like forgetting, not memory loss, but a fading of intensity around old priorities. Things you used to care about deeply, competitive success, physical dominance, the need to be seen as capable, they start to feel less central. Not because you've decided they don't matter, but because the neurons that made them feel urgent are operating at a lower amplitude. And maybe that's okay. Maybe the brain is just making room for other things to feel important, things that don't require urgency, things that don't require a surge. The cells themselves don't mourn the change. They don't resist it. They just adapt. Because adaptation is what neurons do. They receive input. They adjust their structure. They keep firing, even when the input changes, even when the chemical environment shifts, even when the molecules that once defined their behavior start to arrive less often. The memory these cells hold isn't narrative. It's structural. It's in the thickness of a dendritic spine, in the clustering of receptors at a synapse, in the speed at which an action potential travels down an axon. And as testosterone continues to fall, these structural memories begin to shift. The neuron that once fired rapidly in response to challenge starts to fire more slowly. The neuron that once released a flood of dopamine starts to release a trickle. The neuron that once amplified the feeling of wanting starts to settle into a quieter baseline. And somewhere inside you, without ever being told, you begin to want things differently. Not less, just differently. The cells don't know what this means for you. They only know what it means for them. Less androgen activation, adjusted gene expression, recalibrated dopamine output, a new equilibrium forming in the space where the old one used to be. And beneath all of this, beneath the receptors and the synapses and the slowly changing firing rates, the question remains. What does a neuron remember when the signal it's been listening to for decades starts to fade? Not the content of motivation, not the reasons, just the shape of it, the rhythm, the intensity it used to carry. And now, in the absence of that intensity, the neuron begins to learn a different rhythm. One that doesn't pulse as hard, one that doesn't demand as much, one that simply continues. There is a gap between neurons, a space so small it exists only in measurements that don't correspond to anything you can picture. And across this gap, dopamine molecules are released, drifting through fluid that fills the emptiness, travelling toward receptors on the other side. And this journey, this crossing from one cell to another, is where transmission happens, where one neuron's electrical impulse becomes another neuron's chemical instruction. And it's not entirely clear whether the speed of this process matters as much as we once thought, or whether the brain simply adjusts to whatever speed it receives, recalibrating its sense of normal around the tempo it's given. When testosterone is present in higher amounts, circulating through the bloodstream and crossing into the brain, it doesn't enter the synapse directly, but it does enter the neurons on either side of it, binding to androgen receptors inside the cell body, inside the nucleus, influencing the production of proteins that will eventually make their way to the synaptic terminal, the place where dopamine is stored in tiny vesicles, waiting for the signal to be released. And when testosterone levels are sustained, these proteins are produced in quantities that allow for robust transmission, for quick reloading of dopamine after each release, for efficient reuptake and recycling, for a system that operates at high capacity, not because it's straining, but because that's simply the baseline it's been maintaining. But as testosterone declines, not suddenly, not in a way that triggers any alarm within the cells themselves, the production of these synaptic proteins begins to slow. The enzymes responsible for synthesizing dopamine are expressed at lower levels. The vesicles that package dopamine may become slightly less numerous or slightly less full. The transporters that pull dopamine back out of the synapse after it's been released, recycling it for future use, may become less efficient. And the cumulative effect of all these small changes is that transmission across the synapse begins to happen at a different rate. Not absent, not broken, just slower, softer, less intense. It's a bit like watching a river that's been fed by heavy rains gradually return to its regular flow. Not dry, not stopped, just moving with less force, carrying less volume, and the riverbed remains the same, The path is unchanged, but what travels through it arrives on the other side with less momentum, and the landscape downstream adjusts to the new conditions without fanfare, without resistance, because this is simply what water does when the source diminishes. The synapse itself is a remarkably patient structure, built to endure constant activity, constant chemical flux, constant adjustment, and it doesn't interpret the reduction in dopamine release as failure. It interprets it as information, as a new set of conditions to accommodate. And so the postsynaptic neuron, the one receiving the dopamine signal, begins to modify its surface, increasing or decreasing receptor density, depending on what seems most adaptive, trying to maintain some level of functional response even as the input changes. And this process, called synaptic plasticity, happens without your awareness, without your consent, happening in the background of your life while you sleep, while you work, while you wonder why certain things don't feel as compelling as they used to. There's something we still don't fully understand about how quickly these adaptations happen, whether the brain compensates faster in some people than others, whether age influences the speed of synaptic remodeling, whether the decline in testosterone itself triggers protective mechanisms that soften the transition, or whether the system simply drifts into a new equilibrium without much resistance, accepting the change as inevitable, as part of the ongoing recalibration that defines aging at the cellular level. What we do know is that the dopamine transporter, the protein responsible for clearing dopamine out of the synapse after it's been released, is itself influenced by androgens, and when testosterone levels drop, the expression of this transporter can change, sometimes decreasing, which means dopamine lingers in the synapse slightly longer, compensating somewhat for the reduced release, or sometimes remaining stable, which means the synapse clears at the same rate even though less dopamine is arriving, and the net effect depends on which change dominates, which adaptation the neuron prioritizes, and this varies, not just between people, but between different synapses within the same brain, creating a mosaic of transmission speeds, some slower, some unchanged, some quietly compensating. The neurons don't communicate with each other about this. They don't coordinate their responses, they simply respond to their local environment, to the chemical signals they receive, to the presence or absence of testosterone binding to androgen receptors, to the concentration of dopamine in the synapse, to the frequency of action potentials arriving from upstream, and each synapse adjusts independently, becoming a slightly different version of what it was, and across thousands of synapses, across entire neural circuits, these small independent adjustments accumulate into something that feels, from the inside, like a shift in how motivation works, how pleasure registers, how desire pulls you forward or doesn't. You could think of it as the difference between a shout and a whisper, though that's not quite right because the message itself hasn't changed. Only the volume, only the urgency with which it's delivered, and the brain on the receiving end learns to listen more carefully, to attend to signals that used to be louder, to extract meaning from transmissions that arrive with less force. And in this way, the system doesn't fail. It adapts. It continues. It finds a way to keep functioning, even as the neurochemical context shifts beneath it. The vesicles themselves, those small membrane-bound spheres that store dopamine inside the presynaptic terminal, are not passive containers. They are regulated by proteins whose expression depends partly on androgenic signaling, and when testosterone falls, the number of vesicles ready to release dopamine at any given moment may decrease, or the amount of dopamine packed into each vesicle may drop, or both. And this means that when an action potential arrives at the terminal, when the signal comes to release dopamine into the synapse, what gets released is a smaller packet, a quieter message, and the postsynaptic neuron receives this quieter message and must decide how to respond, whether to amplify its sensitivity, or to accept the reduction as the new baseline. There's a certain elegance to how this happens, a kind of biological grace in the way the system doesn't collapse, but simply reconfigures, finding new points of stability, new operating ranges, and it's not entirely clear whether this reconfiguration preserves function perfectly or whether something is inevitably lost in the transition, whether the brain can fully compensate for reduced dopamine signaling, or whether some experiences, some aspects of motivation and reward, become genuinely harder to access, not because the pathways are gone, but because the signal travelling through them is too faint to register with the same intensity. The synapse continues, the neurons continue, the transmission continues, but it continues differently, in a lower key at a slower tempo, and whether this constitutes loss or simply change depends on what you are relying on that dopamine surge to provide. What parts of your life were built around the assumption that motivation would always arrive with force, that desire would always feel urgent, that the chemical systems underwriting your sense of drive would remain consistent across decades? They don't remain consistent, they adapt, they recalibrate, they slow without stopping, and the synapse, that tiny gap where one neuron speaks to another, becomes a place where transmission still happens, just more quietly, more patiently, as if the brain has decided that urgency was optional all along, that the message can still be delivered even when it arrives without the sharpness it used to carry, even when the dopamine molecules drift across the gap more slowly, even when the receptors on the other side receive fewer of them and must work harder to translate those fewer signals into something meaningful. And so the transmission continues, slower, softer, but not stopped, not broken, just moving through a synapse that has learned to operate under different conditions, under the quiet, steady influence of declining testosterone, under the gradual rewriting of what transmission means when the molecules that once amplified it are no longer present in the same concentrations, and the brain, patient and adaptive, continues to send messages across these gaps, continues to build motivation and desire and reward out of whatever dopamine is available, out of whatever signal strength remains. The gap doesn't widen, but the crossing takes longer, and on the other side the neuron waits, still ready, still listening, learning to hear what arrives, even when it arrives more faintly, even when the transmission feels less like a declaration and more like a murmur, something still worth attending to, something still capable of meaning. There was a time years ago when this system worked like a thermostat. The brain detected a need, the body responded. Testosterone rose or fell based on signals from the hypothalamus, that small regulatory center deep in the brain. Dopamine activity shifted in response to both the testosterone itself and to the experiences testosterone helped amplify. And then the brain measured the results, adjusted its next instruction, maintained equilibrium. Back and forth, signal and response, a closed loop. That's how feedback systems work when all the components are listening. But something has changed in the listening. The hypothalamus still sends its signals. Gonadotropin-releasing hormone still pulses from its neurons, travelling down to the pituitary gland, instructing it to release luteinizing hormone into the bloodstream. Luteinizing hormone still reaches the testes, still binds to Leydig cells, still triggers testosterone production. The machinery is intact, but the output has declined. The Leydig cells, after decades of responding to the same signals, have become less responsive. They produce less testosterone per unit of luteinizing hormone. The hypothalamus compensates by increasing its signaling. The pituitary releases more luteinizing hormone. But even with higher stimulation, testosterone production remains lower than it was. This is not a broken thermostat. This is a thermostat recalibrating around a colder room. And the dopamine system, which has been part of this conversation all along, begins to adjust to the information it's no longer receiving. Testosterone used to arrive at dopamine neurons with regularity. It crossed the blood-brain barrier, diffused through neural tissue, bound to androgen receptors inside cells. Those receptors, once activated, moved into the cell nucleus and influenced gene transcription. Proteins were built, enzymes were produced. Dopamine synthesis increased. The presence of testosterone told dopamine neurons to operate at a certain capacity. Now that presence is diminished. The androgen receptors still exist. They still function. But they spend more time unoccupied. And an unoccupied receptor sends no signal. Silence is also information. The dopamine neurons interpret this silence as instruction. They reduce the production of tyrosine hydroxylase, the rate-limiting enzyme in dopamine synthesis. They decrease the number of vesicles loaded with dopamine. They lower their baseline firing rate. The feedback loop hasn't broken. It has simply stopped receiving one of its inputs. Imagine a conversation between three people. One person stops speaking, the other two continue talking, but the absence changes the discussion. Certain topics no longer come up. Certain perspectives are missing. The conversation continues, but it drifts. That's what's happening between the hypothalamic pituitary gonadal axis and the dopaminergic system. The hormonal axis keeps running its cycle. GnRH pulses, LH rises. Testosterone is produced, just less of it. The axis receives feedback from the lower testosterone levels and tries to compensate, but the compensation is incomplete. The system settles at a new baseline, and dopamine neurons, which were never formally part of this endocrine loop but were always influenced by it, recalibrate around the absence. They don't stop producing dopamine, they produce it in response to different priorities now. There are other neuromodulators, serotonin, norepinephrine, endogenous opioids. These systems begin to contribute more to the regulation of mood and motivation. They pick up some of the slack, not all of it, but some. The brain is solving for equilibrium again, just with different variables. Testosterone used to weight certain experiences more heavily. Competition, physical achievement, assertive social behavior. The dopamine released in response to these experiences was amplified by androgenic signaling. The reward felt stronger. The motivation to repeat the behavior was higher. Without that amplification, the dopamine response to these experiences becomes more neutral, not absent. Neutral. You still feel satisfaction from achievement, but the feeling doesn't surge the way it used to. It registers. It passes. You move on. And the feedback loop, which once reinforced these behaviors strongly, now reinforces them moderately. The brain still learns. It still updates its predictions about what's rewarding. It just updates them based on a quieter signal. The hypothalamus monitors this indirectly. It doesn't have direct access to dopamine levels, but it does track overall arousal, overall engagement, overall sense of well-being. And if those metrics drop too low, it can attempt further adjustments. More GnRH, more LH. But the Leydig cells have limits. They can only produce so much testosterone in response to stimulation. And those limits become more pronounced with age, so the loop continues, but it operates within tighter constraints. There's a certain resignation in this process. Not emotional resignation, biological resignation. The acceptance of limits. The body has always had limits. It's just that some limits weren't apparent until now. Testosterone production was never infinite. Dopamine signaling was never unconditional. The feedback systems maintaining them were always approximations, always imperfect, always vulnerable. The degradation that comes with time. And now that degradation is expressing itself as a slow drift away from the parameters that defined earlier decades. The loop still runs, hormones still cycle, dopamine still fires, feedback still adjusts output. But the conversation has changed. One voice is quieter, the others compensate. The system finds a new balance, and somewhere in that balance, in the space between signal and response, your sense of what drives you begins to shift. Not because you've chosen it, not because you've lost anything essential, but because the biological dialogue that underlies motivation has reorganized itself around a missing participant. The thermostat still works, it's just maintaining a different temperature now. The hypothalamus still regulates, the pituitary still responds, the gonads still produce, dopamine neurons still fire, everything still functions, just quieter, just less amplified, just operating within the reality of what the Leydig cells can provide and what the dopamine system can sustain without the androgenic input it once relied on. The loop closes, the loop opens, the loop adjusts, and the adjustment continues slowly. Across months, across years, as testosterone continues its gradual decline and dopamine recalibrates again and again, finding new baselines, new equilibria, new ways to generate motivation from a quieter signal. The feedback doesn't fail, it just learns to solve for different outcomes. Outcomes that don't require the intensity testosterone once provided. Outcomes that emerge from steadier, softer neurochemical processes. Outcomes that feel less urgent but no less real. The loop remembers what it used to optimize for, but memory doesn't stop recalibration. It just makes the adjustment feel like loss, even when it's only change. The hypothalamus sends its signal, the pituitary responds, the gonads produce what they can. Dopamine fires in response to what arrives, and the loop, ancient and automatic, continues to run, solving for balance in a system that no longer provides the same inputs it once did, finding equilibrium in the quiet space where urgency used to be. You can't point to it, can't feel it directly, but it's there, right now, tucked behind your eyes, beneath the thalamus, buried in tissue you'll never consciously access. The ventral tegmental area, VTA, a cluster of perhaps 50,000 neurons, maybe fewer. The number varies. The exact count has never seemed to matter as much as what these neurons do. They make dopamine, not all of them. Some produce GABA, the brain's primary inhibitory neurotransmitter. Some produce glutamate. But the dopamine neurons here are the ones that matter for this story. They're the ones that project to the nucleus accumbens, to the prefrontal cortex, to regions involved in reward and motivation and the feeling that something out there is worth pursuing. And scattered throughout these dopamine neurons are androgen receptors, small proteins embedded in the cytoplasm, waiting for testosterone to arrive. They've been waiting your entire life. For decades, testosterone crossed the blood-brain barrier, drifted through extracellular fluid, entered these neurons, bound to these receptors. The receptor-testosterone complex moved into the nucleus. Genes activated, proteins formed. The neuron adjusted its behavior based on this hormonal instruction. It's a quiet instruction. Not dramatic, just persistent. Testosterone told the neuron to maintain a certain level of tyrosine hydroxylase, the enzyme that converts tyrosine, an amino acid from your diet, into L-dopa, which then converts into dopamine. Testosterone influenced the neuron's metabolic rate, its energy consumption, the density of its dendritic spines, those small protrusions where the neuron receives input from other cells. Testosterone didn't create motivation, but it shaped the neuronal infrastructure that makes motivation possible, and now, less testosterone arrives. The androgen receptors wait longer between activations. The genes they regulate receive fewer signals to turn on. The proteins those genes encode are produced in smaller quantities. And the neuron slowly begins to operate differently. It's strange to think about, that a cell you've never felt in a region you've never seen has been listening to a hormone you didn't consciously produce, and that this hormone has been influencing whether you felt driven or indifferent, focused or scattered, hungry for achievement or content to let things be. The dopamine neurons in the VTA don't fire randomly. They fire in response to prediction errors. When something good happens that you didn't expect, they fire rapidly. A burst. When something good fails to happen when you expected it, they pause. A dip. When something happens exactly as predicted, they fire steadily. No change. This is how the brain learns. By tracking the difference between expectation and reality, and testosterone has been modulating the intensity of these signals. With higher testosterone, the burst is stronger. The dip is deeper. The neuron's response to unpredicted reward is more pronounced. With lower testosterone, the bursts are smaller. The dips are shallower. The neuron's sensitivity to prediction error decreases. You learn the same lessons. You just learn them with less emotional weight. A success still registers as success, but it doesn't surge through you the way it used to. A failure still registers as failure, but it doesn't hollow you out the way it might have before. The VTA is recalibrating its teaching signal, and downstream regions, those areas receiving dopamine projections from the VTA, begin to learn differently. The nucleus accumbens, which translates dopamine into the sensation of wanting, starts to want things less intensely. The prefrontal cortex, which uses dopamine to maintain focus on goals, finds it harder to sustain attention on distant objectives. The amygdala, which associates dopamine signals with emotional salience, begins to mark fewer experiences as deeply important. Not because these regions are damaged, because the signal they're receiving from the VTA has changed. The VTA is still teaching, still broadcasting, still firing in response to reward and punishment. It's just teaching in a quieter voice, and I keep thinking if you could zoom into one of these neurons right now, if you could watch it in real time, what would you see? You'd see an axon extending forward, branching into terminals that release dopamine into thousands of synapses. You'd see a cell body filled with mitochondria, those small energy-producing organelles working constantly to keep the neuron alive. You'd see the nucleus, where DNA coils and uncoils, where transcription happens, where the presence or absence of testosterone-bound androgen receptors determines which genes get read. You'd see fewer androgen receptors occupied than there were 5 years ago. Fewer than 10 years ago, the receptor sits empty, waiting. But testosterone doesn't arrive as often, so the receptor stops influencing gene expression as frequently. And the proteins that depend on that gene expression, enzymes, transporters, structural proteins, begin to decline in concentration. The neuron doesn't die, it adjusts. It reduces its dopamine output to match the hormonal environment it's operating in. And this adjustment, multiplied across thousands of VTA neurons, becomes a system-wide recalibration. Motivation doesn't vanish, it becomes more selective. Reward doesn't disappear, it becomes less intense. The VTA is still waiting, still listening, still responding to the world, but it's responding from a different baseline now. A baseline shaped by the gradual withdrawal of androgenic signaling. A baseline that doesn't prioritize urgency, that doesn't amplify competition, that doesn't surge in response to dominance or status or physical achievement the way it used to. The neurons are patient. They don't resist the change. They don't mourn the loss of testosterone's influence. They simply continue doing what neurons do, firing, releasing neurotransmitter, adjusting gene expression based on available signals, finding equilibrium in whatever chemical environment they're given. And maybe that's all this is. Not a failure, not a decline, just a neuron doing what it's always done, adapting to the hormonal context it exists within. The VTA waits, the androgen receptors wait, the dopamine vesicles wait, fewer in number now, less densely packed. And somewhere in that waiting, in the silence between testosterone pulses that arrive less frequently, in the space where activation used to happen and now doesn't, the neuron learns a different rhythm, a quieter one that doesn't depend on androgenic amplification, one that generates motivation from steadier, softer sources, one that continues even in the absence of the hormone that once defined its intensity. The VTA doesn't stop, it waits. And in waiting, it becomes something slightly different than it was. Still functional, still necessary, still capable of generating the dopamine that underlies every goal, every desire, every pursuit. Just doing so with less force, less urgency, less of the sharpness testosterone once provided. The neurons fire, the dopamine releases, the signal travels forward into the brain's reward circuitry, carrying information about what matters and what doesn't, about what's worth pursuing and what can be let go. And in the quiet dimness of the VTA, in the space where androgen receptors sit waiting for a signal that arrives less often now, the system continues, adapting, recalibrating, still teaching the brain what to want, just teaching it more softly. There are parts of the dopamine system that don't care about testosterone at all. They never did. The neurons that respond to food when you're genuinely hungry, the ones that fire when you see someone you love. The circuits involved in curiosity about how something works, the pathways that register the satisfaction of solving a problem. These remain largely intact. They don't rely on androgens to function. They were built on different foundations. So when testosterone declines, these experiences don't fade. You still enjoy food, you still feel affection, you still find things interesting. What changes is everything else. The things that were amplified by testosterone's presence. The desire to win, the need to prove capability, the pull toward physical competition, the edge that made certain achievements feel essential rather than optional. These begin to quiet, not all at once, but steadily, like a radio slowly losing signal strength in certain frequencies while others come through clearly. What stays is the core of dopamine's function: pleasure, learning, attention. What fades is the amplification, the intensity, the urgency, the sense that some goals matter more than survival itself. You can still set goals, you can still work toward them, but the internal pressure to do so, the feeling that you must, that your worth depends on it, that pressure softens. Some people describe this as relief, some describe it as loss, most describe it as both, depending on the moment. The part that feels most fragile is the motivation tied to status, to being seen as strong, to being seen as capable, to being seen at all. Testosterone didn't create these needs, but it made them feel urgent. It turned social comparison into a dopamine-driven process. It made winning feel like the only outcome worth pursuing. And now, without that amplification, social competition starts to feel optional. You notice someone doing better than you. You register it. You don't feel the same compulsion to surpass them. Not because you've given up, because the dopamine response to that comparison is quieter. The burst that used to fire when you imagined overtaking them, it's smaller now. The neural teaching signal is weaker. Your brain is learning that this particular form of achievement doesn't warrant the same investment. And maybe that's accurate. Maybe it never warranted it. maybe testosterone was just convincing you it did. Physical exertion changes too. Exercise still releases dopamine. But the anticipation of it, the drive to push yourself, to test limits, to prove what your body can do, that anticipation is less insistent. You can still choose to exercise. You can still enjoy it, but it doesn't pull at you the way it used to. The dopamine neurons that once fired in anticipation of physical challenge are receiving less androgenic input. They still fire, just with less enthusiasm, less certainty that this is what you should be doing. What remains strong is gentler. Connection. Curiosity without competition. The quiet satisfaction of understanding something. The warmth of being around people you trust. These dopamine pathways were never testosterone dependent. They were built on different neuromodulators: oxytocin, serotonin, endogenous opioids. These systems are still intact, still functioning, still capable of generating pleasure and motivation. Just not the urgent, sharp-edged kind, the softer kind, the kind that doesn't demand, the kind that simply is. You might find yourself drawn to different activities now, not because you've consciously decided to change, but because your dopamine system is reinforcing different behaviors. Long conversations instead of competitive games, observing instead of dominating, appreciating instead of acquiring. The neurons are still learning, still tracking what brings reward. They're just learning from a quieter teaching signal, one that doesn't prioritize dominance, one that doesn't amplify achievement above all else, one that allows other values to surface, values that were always there, just overshadowed. There's a certain gentleness to this process, a softening, not weakness, just less rigidity, less insistence that things must be a certain way. The dopamine pathways tied to testosterone were never the whole story. They were just the loudest part, the most insistent, the most impossible to ignore. And now, as they're quiet, the other pathways become more audible, the ones that never needed amplification, the ones that operate on steadier, quieter principles, the ones that generate satisfaction without urgency. You still feel desire, you still feel pleasure, you still feel motivation, just not always for the same things, and not always with the same intensity. The edge is gone. What remains is softer, steadier, less prone to surge and crash, less dependent on external validation, less convinced that winning is the only outcome worth pursuing. The dopamine is still there, still firing, still teaching your brain what matters. It's just teaching different lessons now. Lessons about patience, about presence, about satisfaction that doesn't require dominance, about pleasure that doesn't need to be earned through competition. These lessons were always available. Testosterone just made them harder to hear. And now, in its absence, in the quiet space where urgency used to be, these gentler teachings begin to surface. Not as replacement, not as compensation, just as what was always there, waiting beneath the noise, waiting for the amplification to fade, waiting for the dopamine system to recalibrate around something steadier than the need to win. What stays is not less than what fades. It's just different, quieter, less insistent on its own importance. And in that quietness, in the space where urgency used to drive every decision, something else emerges, not new, just finally audible, finally able to shape behavior without competing against the sharp-edged intensity testosterone once provided. The dopamine continues, the learning continues, just toward different ends now, softer ones, steadier ones, ones that don't require you to prove anything at all. The receptor doesn't think, it doesn't decide, it just waits. A protein structure folded into a specific shape positioned in the cell membrane where the outer environment meets the inner machinery of the neuron, and when dopamine arrives, when a molecule drifts across the synapse and binds to this receptor, the protein changes shape, triggers a cascade of events inside the cell, opens ion channels or activates second messengers or influences gene expression, depending on which type of receptor it is. There are 5 main types of dopamine receptors, D1 through D5. Each one does something different when activated. D1 and D5 tend to excite the neuron, making it more likely to fire. D2, D3 and D4 tend to inhibit, making it less likely to fire. The balance between these receptors determines how a neuron responds to dopamine, and testosterone influences that balance, not directly but through the regulation of genes that control receptor expression. And when testosterone levels are high, certain dopamine receptors are produced in greater numbers, particularly D2 receptors in regions like the striatum and nucleus accumbens. And this higher density of receptors means that even moderate amounts of dopamine can produce a strong response, can trigger significant changes in neuronal activity, can translate into noticeable shifts in motivation or pleasure or attention. But when testosterone falls, the expression of these receptors begins to change. The cell reduces production. In some neurons, D2 receptor density drops. The same amount of dopamine now binds to fewer receptors. The response is weaker. It's not entirely clear why the brain makes this adjustment, whether it's an attempt to conserve resources or simply a passive consequence of reduced androgenic signaling. But the effect is measurable, observable in animal studies, where testosterone is experimentally lowered and receptor density is tracked over time. Fewer receptors means the neuron is less sensitive to dopamine. A surge that once felt significant now barely registers. The neuron requires more dopamine to produce the same level of activation it used to achieve with less, but dopamine production is also declining, so the system is adjusting downward on both ends, both in the signal sent and in the ability to receive it, and this dual reduction compounds the overall effect, making the experience of reward and motivation noticeably quieter. The architecture of want is built on these receptors. When you desire something, when anticipation builds, when the thought of achieving a goal produces a feeling of pull, dopamine is binding to receptors in your nucleus accumbens, in your prefrontal cortex, in your striatum. The more receptors available, the stronger that pull feels. The fewer receptors, the more diffuse the sensation becomes. You still want things, but the wanting feels less urgent, less consuming, more like a preference than a need. And this change happens gradually, across months and years, as testosterone continues its slow decline, and the genes regulating receptor expression respond to the shifting hormonal environment. The neuron doesn't fight this process. It adjusts receptor density based on the signals it receives. If dopamine arrives less often, or in smaller amounts, the neuron may upregulate receptors to compensate, growing more of them to catch every available molecule. But if androgenic signaling is also declining, the instruction to produce receptors weakens, and upregulation becomes incomplete or doesn't happen at all. The neuron settles at a lower density. It accepts this as the new baseline, and the result is a brain that experiences dopamine-driven processes with less intensity than before, not because dopamine itself has vanished, but because the machinery for detecting and responding to it has been scaled back. Some receptors aren't affected by testosterone at all. D1 receptors, for instance, seem relatively stable across changes in androgen levels, at least in some brain regions. They continue to respond to dopamine the way they always have, but D2 receptors, which are heavily involved in motivation and reward prediction, show clear sensitivity to testosterone, and when testosterone drops, D2 receptor density follows. This creates an imbalance in the dopamine system. Excitatory signaling through D1 receptors remains relatively intact while inhibitory signaling through D2 receptors weakens, and it's not entirely clear what this imbalance does to overall brain function, whether it shifts the system toward more excitation or whether other compensatory mechanisms kick in to restore equilibrium. What we do know is that motivation changes. Goal-directed behavior becomes harder to sustain. The internal sense that a particular outcome matters diminishes, and this seems to correlate with reduced D2 receptor availability in key motivational circuits. The receptor sits in the membrane, it waits for dopamine. When dopamine arrives, it binds, the receptor activates, the neuron responds. But if there are fewer receptors waiting, if the density has dropped by 10%, 20%, 30% over the course of years, then the same dopamine release produces a proportionally weaker response. The neuron fires less vigorously, the downstream effects are blunted, the behavior motivated by that dopamine signal becomes less compelling. And you experiencing this from the inside might interpret it as apathy, or fatigue, or simply getting older, not realizing that the change is structural, cellular, happening at the level of protein expression in neurons you'll never consciously access. There's something almost quiet about how this happens. No inflammation, no cell death, no dramatic failure, just a slow reduction in receptor density, just a gradual recalibration of how sensitive the brain is to its own dopamine. The architecture of want doesn't collapse, it becomes less responsive, less inclined to surge, less capable of translating dopamine into the intense, consuming desire that used to drive behavior. And maybe that's not entirely bad, though it's hard to say, hard to separate the neurochemistry from the life experience. Hard to know whether reduced receptor density is a protective mechanism or simply an inevitable consequence of aging and hormonal decline. The receptors that remain continue to function. They're not damaged, they're not broken, they just exist in lower numbers. And the neuron, operating with this reduced receptor population, continues to respond to dopamine, continues to participate in the circuits of motivation and reward, just with less sensitivity, less responsiveness, less capacity to generate the kind of urgent wanting that once felt like the center of existence. The want is still there. It's just quieter now, built on fewer receptors, sustained by a dopamine system that's learning is to operate within the constraints of declining testosterone. Learning to generate motivation from less, learning to translate smaller signals into meaningful behavior, learning to want things without the intensity that used to make wanting feel like necessity. The architecture adapts, the receptors thin out, the brain recalibrates. And somewhere in that recalibration, in the space between what the system used to be capable of and what it can sustain now, the experience of desire shifts into something gentler, something that doesn't demand as much, something that allows you to want without being consumed by the wanting. The receptors wait, fewer of them now, spaced further apart on the membrane, still capable of binding dopamine when it arrives, still capable of triggering a response, just a quieter one, a response that doesn't fill the entire neuron with urgency, that doesn't translate into behavior with the same force it once did. The architecture of want remains, just built on a thinner foundation, just operating within the reality of what fewer receptors can provide. There's a region in your brain shaped like a comma, curved and dense with cells, sitting just beneath the cortex where conscious thought happens, and this region doesn't care about your intentions or your reasoning, or the stories you tell yourself about why you do what you do. It only cares about patterns, about what worked before, about the feeling that follows an action, and whether that feeling was pleasant enough to store the action as something worth repeating. And this is the striatum, a structure so old in evolutionary terms that versions of it exist in nearly every vertebrate brain, refined across millions of years to do one thing exceptionally well: learn from consequences and shape future behavior accordingly. The striatum is where habits live, where the automaticity of reaching for your phone without thinking begins, where the sequence of movements required to tie your shoes or drive a familiar route gets encoded so deeply that consciousness barely participates. And it's also where reward is processed, where dopamine arriving from the ventral tegmental area and Substantia nigra tells the striatum whether the action you just took led to something desirable or something neutral, or something you'd rather avoid next time, and over years, over decades. This constant feedback loop between action and dopamine response builds a vast internal library of behaviors ranked by how rewarding they've been, how worth repeating they seem to be. Testosterone has been part of this process from the beginning, not by creating the striatum's function, but by modulating it, by influencing how strongly dopamine signals register here, by affecting receptor density and neurotransmitter release and the excitability of the medium spiny neurons that make up most of the striatum's cellular architecture. And when testosterone was higher, the striatum operated with a particular sensitivity, a particular responsiveness to reward that made certain behaviors, competitive ones, physically demanding ones, status-seeking ones, feel especially worth encoding as habits, as patterns to repeat. But now testosterone is lower, and the striatum, patient and mechanical in its function, begins to recalibrate what it considers rewarding, what it bothers to encode as worth doing again. It tends to think, though thinking isn't quite the right word for what the striatum does, it tends to seem like the striatum is conservative in its preferences, like it favours the familiar over the novel, the established pattern over the untested action. And this makes sense because the striatum's job is efficiency, is automating behaviours so the cortex doesn't have to consciously manage every small decision, and testosterone used to make certain novel behaviours feel rewarding enough to overcome that conservative bias, used to provide enough dopamine-driven reinforcement that trying something new, taking a risk, asserting yourself in unfamiliar territory felt worth the effort, felt like it might lead to outcomes the striatum would want to remember. Without that reinforcement, the striatum becomes even more conservative, even more inclined to stick with what's already been proven safe and minimally rewarding, rather than expending energy on actions that might or might not pay off. And this isn't a conscious choice, isn't something you decide. It's just the striatum doing what it's always done, adjusting its learning rate based on the dopamine feedback it receives, and with less dopamine arriving per reward, with weaker signals telling it that something was worth doing, the threshold for encoding a new habit rises. The bar for what counts as rewarding enough to repeat gets higher. You might notice this as a reluctance to start new routines, a preference for the familiar even when the familiar isn't particularly satisfying, a sense that it takes more effort to build momentum towards something you haven't done before. And these aren't failures of willpower or discipline. They're the striatum operating under different neurochemical conditions, responding to a dopamine system that's been recalibrated by the absence of testosterone's amplifying influence. The medium spiny neurons that populate the striatum are densely packed with dopamine receptors, particularly D1 and D2 receptors, and these receptors determine whether a neuron becomes more or less likely to fire in response to a given input, whether it strengthens or weakens the connection between a cue and an action, and testosterone affects the expression and sensitivity of these receptors, meaning that as testosterone declines, the striatum's ability to form strong associations between actions and rewards diminishes slightly, not catastrophically, just enough that behaviors need to be more clearly rewarding, more obviously beneficial, before the striatum bothers to encode them as worth automating. There's a subdivision within the striatum called the dorsal striatum, involved primarily in habit formation and motor control, and another called the ventral striatum, which includes the nucleus accumbens and is more involved in motivation and reward anticipation. And both of these regions contain androgen receptors, both respond to testosterone, both recalibrate when testosterone levels fall, though they recalibrate in slightly different ways. The dorsal striatum seems to become less flexible, less willing to update old habits or form new ones, while the ventral striatum becomes less sensitive to anticipated rewards, less inclined to generate the motivational pull that makes you want to pursue goals in the first place. If you imagine the striatum as a kind of voting system where different patterns of activity compete to determine which behavior gets expressed, which habit gets activated, which goal gets pursued, then testosterone was like a bias that weighted certain votes more heavily. Votes for competitive behavior, for physical exertion, for status-driven action. And without that bias, the voting becomes more evenly distributed, less skewed toward any particular category of behavior. And what emerges is a striatum that treats most options as roughly equivalent unless one stands out as exceptionally rewarding or exceptionally familiar. And since most new behaviors don't stand out that way, since most goals require sustained effort before they pay off, the striatum increasingly defaults to whatever's easiest, whatever's already encoded, whatever requires the least dopamine investment to execute. This is why habits become harder to change with age, why motivation feels less automatic, why the things you used to pursue with energy now require conscious effort to initiate. And it's not because the striatum has stopped working, it's because the striatum is working exactly as it should, given the dopamine environment it's operating within, prioritizing efficiency over exploration, familiarity over novelty, conservation over risk. The recalibration happens slowly, across years, as testosterone continues its decline and dopamine signaling adjusts in response, and the striatum, interpreting this new baseline as simply the reality it needs to work within, begins to favor patterns that don't require strong dopamine reinforcement to sustain, patterns that can run on minimal reward, patterns that have already been encoded deeply enough that they persist, even when the dopamine surge that created them has faded. And maybe this is fine. Maybe the striatum's increasing conservatism is adaptive in its own way, a recognition that energy is finite, that not every goal is worth pursuing, that some behaviors can be let go without loss. But it does mean that the sense of momentum you used to feel, the ease with which new habits formed and old ones updated, the spontaneity of trying something just to see if it might be rewarding, all of that becomes harder to access, not impossible, just requiring more conscious effort than it used to, more deliberate override of the striatum's preference for what's already known. The striatum doesn't announce these changes, doesn't send signals to consciousness explaining that it's recalibrating, that it is adjusting its learning algorithms based on reduced dopamine input. It just quietly shifts the probabilities, makes certain behaviors slightly more likely and others slightly less, and you experience this as a change in what feels natural, what feels worth doing, what pulls at you enough to overcome inertia. The patterns are still there. The capacity for habit and reward and motivated action still exists. The striatum still functions, still learns, still shapes behavior based on dopamine feedback. It's just doing so within a narrower range now, with less sensitivity, less willingness to encode new patterns unless they're clearly, obviously, undeniably worth the effort. And most things aren't that clear. Most rewards are subtle and delayed and uncertain, and so the striatum, faced with uncertainty, defaults to what it already knows, to the habits it's already built, to the behaviors that require the least cognitive and neurochemical investment to execute. And in that defaulting, in that quiet retreat toward the familiar, the striatum becomes less a driver of change and more a guardian of stability, less a system pushing you toward new goals and more a system maintaining whatever equilibrium you've already established. And whether that's loss or simply transformation depends on what you needed the striatum to do, whether you needed it to keep pushing you toward achievement or whether you needed it to finally let you rest. The striatum recalibrates, it doesn't stop, it just learns to work with less dopamine, less testosterone, less reinforcement for novelty, and in working with less it becomes more selective, more conservative, more inclined to favour the known over the unknown. The patterns it's already built remain. The new patterns it might build become harder to form, and somewhere in that shift, movement and habit and reward all begin to feel like they require more effort than they used to. Not because anything is broken, but because the striatum is simply doing what it's always done, optimizing for efficiency within whatever neurochemical context it's given. 10 years ago, the synapses in your striatum looked different than they do now. Not dramatically, not in ways that would show up on any scan you could take, but at the molecular level, in the precise arrangement of proteins that hold each connection together, in the density of receptors clustered at the postsynaptic membrane, in the number of vesicles waiting at the presynaptic terminal ready to release dopamine when the next action potential arrives. Back then, testosterone was crossing into your brain at higher concentrations, binding to androgen receptors inside neurons, influencing the expression of genes that code for synaptic scaffolding proteins, those structural molecules that determine how stable a synapse is, how easily it can be strengthened or weakened, how much neurotransmitter it releases per signal, and under the influence of sustained androgenic signaling, your dopamine synapses were configured for high-capacity transmission, built to carry intense signals, optimized for the kind of sharp, urgent motivation that testosterone-enhanced dopamine activity produces. But synapses are not permanent structures in the way bone or muscle tissue is permanent. They are dynamic, constantly being remodeled in response to activity and to the chemical environment they exist within. And every night while you sleep, while consciousness dims and the brain shifts into maintenance mode, synaptic remodeling happens. Connections that were used frequently during the day get strengthened, proteins are synthesized and transported to the synapse to reinforce it, while connections that went unused get weakened. Proteins are removed, the synapse shrinks slightly or disappears entirely if it's no longer serving a function. This process is called synaptic plasticity, and it's how the brain adapts to experience, how it learns, how it updates its internal model of the world based on what happened today and what might happen tomorrow. And testosterone has always been part of the chemical context that guides this remodeling, influencing which synapses get prioritized for strengthening, which connections the brain decides are worth maintaining at high capacity. Now that testosterone is lower, the remodeling process continues, but it's guided by different chemical instructions, by a hormonal environment that no longer emphasizes the same priorities it used to, and the synapses in your dopamine pathways begin to reshape themselves accordingly, not all at once, not in a way that announces itself, but gradually, across months and years, as each night's remodeling makes small adjustments that accumulate into structural change. The scaffolding proteins that hold a synapse together, proteins with names like PSD95 and Homer and Shank, are partially regulated by androgen signaling, and when testosterone levels drop, the expression of some of these proteins decreases, meaning that synapses have less structural support than they used to. They become slightly less stable, slightly more vulnerable to weakening if they're not being used frequently. And in dopamine pathways that were once highly active, that once carried constant signals related to competition and achievement and physical drive, the reduced activity combined with reduced structural support means these synapses begin to shrink. It's a bit like a hiking trail that used to see heavy foot traffic but now only gets walked occasionally. The path is still there, still visible, still usable, but it's narrower than it was, overgrown in places, requiring more effort to navigate because the constant use that kept it clear has diminished. And the synapses in your brain undergo something similar, not eroding entirely but becoming less prominent, less efficient, less capable of transmitting signals with the speed and intensity they once managed. You'll never feel this happening, never notice the moment when a particular synapse loses 10% of its scaffolding proteins, or when the number of dopamine receptors at a postsynaptic membrane drops from 200 to 180. These changes occur at scales far below conscious awareness, in spaces measured in nanometers, but the cumulative effect of thousands of synapses remodeling in response to reduced testosterone does eventually register as something you can feel, as motivation that takes longer to build, as pleasure that feels less intense, as goals that seem further away than they used to. The dendritic spines, those small protrusions on a neuron's dendrites where most excitatory synapses form, are particularly sensitive to hormonal changes, and testosterone influences their density and their morphology, the exact shape they take, and when testosterone declines, some dendritic spines begin to retract, pulling back into the main body of the dendrite, and the synapses that formed on these spines are lost, not because the neuron is damaged, but because the chemical environment no longer supports maintaining that many connections, and the brain, always economizing, always trying to operate efficiently within whatever resources are available, prunes away the connections it can no longer justify energetically. This pruning is selective, it doesn't happen randomly. Synapses that are still being used frequently, that still receive strong input, that still participate in active neural circuits, these tend to be preserved even as testosterone falls, but synapses that were only active when testosterone was amplifying certain motivations, synapses involved in status-seeking behavior or competitive drive, or the pursuit of dominance. These receive less input now, and with less input and less hormonal support, they become targets for removal during nightly remodeling. The brain doesn't interpret this as loss, it interprets it as optimization, as clearing away connections that are no longer serving the organism's current needs. And in a way, this is adaptive. This allows the brain to reallocate resources towards synapses that are still active, toward connections that support behaviors that remain rewarding even without testosterone's amplification. But from the inside, from the perspective of someone experiencing the effects of this remodeling, it can feel like parts of yourself are fading, like desires that used to define you are becoming optional, like the person you were is slowly being replaced by someone quieter, less driven, less certain about what's worth pursuing. At the presynaptic terminal, where dopamine is released, the number of active zones, specialized sites where vesicles dock and fuse with the membrane to release neurotransmitter, can decrease when androgenic signaling weakens, meaning that even if a neuron fires an action potential with the same frequency it always has, the amount of dopamine released per action potential is lower, because there are fewer sites capable of releasing it. And this reduction in release capacity combines with reduced receptor density on the postsynaptic side to create a synapse that transmits signals at significantly lower amplitude than it did when testosterone was higher. The remodeling continues every night. It's not a one-time event but an ongoing process, and as long as testosterone remains low, the synapses will continue to adapt to that reality, will continue to reshape themselves around reduced androgenic input, and over time this creates a dopamine system that is fundamentally different in its architecture, not in the number of neurons or the pathways they form, but in the strength and efficiency of the connections between them, in the capacity of each synapse to transmit information. And perhaps this is necessary. Perhaps the brain recognizes that sustaining high-capacity dopamine synapses requires energy and resources that are better allocated elsewhere when the behaviors those synapses supported are no longer being prioritized. Perhaps synaptic remodeling in response to falling testosterone is the brain's way of accepting a new phase of life. A phase where motivation doesn't need to be urgent, where reward doesn't need to be intense, where the synaptic infrastructure can afford to be leaner, more efficient, optimized for steadier, quieter goals. But it does mean that the brain you have now is not the same brain you had a decade ago, not in its fundamental structure, but in the details, in the fine-grained architecture of its synapses, in the specific configuration of proteins at each connection, and these changes, invisible and unfelt as they occur, accumulate into a system that processes dopamine differently, that responds to reward differently, that generates motivation through connections that have been quietly, gradually, irreversibly remodeled. The synapses reshape. They strengthen where they're needed. They weaken where they're not. And in the spaces between neurons, in the chemical environments that guide each night's remodeling, the absence of testosterone continues to influence which connections survive and which fade, which pathways remain robust and which thin out, and the brain, patient and adaptive, continues its work of rebuilding itself around what's available now, not what used to be, constructing a new architecture of want from synapses that have learned to function with less. Energy is not something neurons can store. They make it as they need it. Every action potential, every neurotransmitter release, every maintenance of membrane potential requires ATP, adenosine triphosphate, the molecule that powers nearly everything a cell does, and ATP is made in mitochondria, those small organelles scattered throughout the neuron's cytoplasm, hundreds of them in a single cell, thousands in some neurons. They've been there your entire life, converting glucose and oxygen into usable energy, running continuously, never stopping, but their output is not fixed. Mitochondria respond to the cellular environment, to available nutrients, to oxidative stress, to hormones, including testosterone. Androgen receptors exist not just in the nucleus, not just in the cytoplasm, but also in mitochondria themselves. Testosterone can bind directly to mitochondrial androgen receptors, influence their function, affect how efficiently they produce ATP. When testosterone levels are higher, mitochondria in dopamine neurons operate at a certain capacity. They produce enough ATP to support high rates of neurotransmitter synthesis, enough to maintain the ion gradients required for rapid firing, enough to power the active transport processes that recycle dopamine after release. The neuron has the energy it needs. It doesn't struggle. But as testosterone declines, mitochondrial function begins to shift. Not dramatically, not in a way that causes immediate cell death or dysfunction, just a reduction in efficiency, a slight decline in ATP output per mitochondrion. You start to notice it indirectly. The neuron that used to fire rapidly now fires more slowly, not because it's lost the ability to fire, because it doesn't have quite as much energy available to sustain rapid firing. The synthesis of dopamine slows slightly. The enzymes involved in converting tyrosine to L-dopa to dopamine require ATP. They require energy cofactors, and when mitochondrial output drops, these processes become slightly less efficient. The neuron can still make dopamine, it just makes it more slowly. And when an action potential arrives, when the signal comes to release dopamine into the synapse, the process of vesicle fusion, of neurotransmitter exocytosis, requires energy, requires ATP to power the molecular machinery that brings vesicles to the membrane and releases their contents. With less ATP available, this process becomes marginally slower, less robust. The neuron still releases dopamine, just less of it per signal, and with slightly more delay. Mitochondria also play a role in calcium handling. Calcium ions flood into neurons during action potentials. They trigger neurotransmitter release. They're essential for synaptic transmission, but calcium needs to be cleared from the cytoplasm quickly after each signal. Too much calcium for too long is toxic to cells. Mitochondria help with this. They sequester calcium, buffer it, keep intracellular levels within safe ranges. When mitochondrial function declines, calcium buffering becomes less efficient. The neuron experiences slightly higher calcium levels after each firing event, not enough to cause damage immediately, but enough to alter cellular processes, to influence gene expression, to potentially accelerate aging at the cellular level. The mitochondria themselves age too. They accumulate oxidative damage over time. Their DNA, which exists separately from nuclear DNA, develops mutations. Their membranes become less efficient at maintaining the proton gradients required for ATP synthesis. And testosterone appears to have some protective effect on mitochondria. It influences the expression of antioxidant enzymes. It supports mitochondrial biogenesis, the process by which new mitochondria are made to replace damaged ones. When testosterone falls, that protection weakens. Damaged mitochondria accumulate. They are not replaced as quickly. The overall mitochondrial population in dopamine neurons becomes older, less efficient, less capable of meeting the neuron's energy demands. This doesn't happen overnight. It happens across years, across decades, but eventually the cumulative effect becomes noticeable. Neurons that once operated with abundant energy now operate closer to their metabolic limits. They still function, they still fire, they still release dopamine, but they do so with less reserve capacity, less ability to respond to sudden increases in demand. And when you try to generate motivation, when you try to pursue a goal that requires sustained dopamine activity, the neurons struggle slightly, not enough to fail, but enough to make the effort feel harder than it used to. The mitochondria are still there, still producing ATP, just less of it, less efficiently. And the neuron, always adapting, always trying to survive within whatever energy budget it's given, begins to reduce its activity to match what the mitochondria can sustainably provide. It fires less frequently, releases less dopamine, scales back its operations to fit within the new energetic constraints. This is part of why motivation becomes harder to sustain with age. Not just because dopamine receptors are changing, not just because testosterone is lower, but because the fundamental energy production system inside dopamine neurons is operating at reduced capacity. The mitochondria can't keep up with the demands they used to meet easily, and the neuron, faced with limited energy, chooses conservation over intensity, chooses to keep functioning at a lower level rather than risk exhaustion. It's a sensible adaptation in many ways. The neuron survives. It continues to participate in neural circuits. It doesn't die, but it does so with less enthusiasm, less capacity for the kind of sustained, energetic activity that characterized earlier decades. The ATP keeps getting made, the glucose keeps getting metabolized, the oxygen keeps being consumed, just at a slower rate, with less efficiency, with more oxidative byproducts that the aging mitochondria struggle to clear. And beneath everything, beneath all the receptor changes and synaptic remodeling and hormonal shifts, this energetic limitation shapes what the dopamine system can actually do, shapes how much motivation it can generate, how much pleasure it can signal, how intensely it can respond to rewards. The mitochondria set the floor, the minimum operating level, and as they age, as their function declines in response to falling testosterone and accumulated damage, that floor rises. The neuron can't operate as far below its maximum capacity as it used to. It has to stay closer to its limits just to maintain basic function. And this feels, from the inside, like everything requiring more effort. Like the reserve tank is smaller. Like pushing yourself is harder. Like sustaining motivation for extended periods drains you in ways it didn't before. Not because you're weaker, not because you've lost anything essential, but because the energy-producing organelles inside your dopamine neurons are quietly, gradually operating at reduced capacity. And the neurons, responding to this reduced capacity, have recalibrated their activity downwards to match what the mitochondria can sustainably provide. The system still works. It just works within tighter energetic limits. Limits set by mitochondria that have been producing ATP without rest for decades and are finally beginning to show the strain. The mitochondria continue. They produce ATP, they buffer calcium, they keep the neuron alive, but they do so with less efficiency now, with less output. And the dopamine neuron, dependent on this energy, adjusts its expectations, reduces its activity, learns to function on less. The energy is still there, just quieter, just enough to keep going, but not enough to surge the way it used to. The mitochondria work, the neuron works, the system works, just closer to its limits now, just with less room for intensity. You've felt this, the effort it takes now to begin something. Not physical effort, mental effort. The work of deciding that something matters enough to focus on. To hold in your attention. To pursue despite everything else competing for awareness. The prefrontal cortex sits just behind your forehead. Managing executive function, planning, decision-making, working memory, the ability to keep a goal active while you work toward it. It does this through dopamine. Dopamine arriving from the ventral tegmental area, binding to D1 receptors in prefrontal neurons, strengthening the signals that represent whatever you're trying to hold in mind. For decades, testosterone modulated this process, not by creating focus, but by amplifying it, by making certain goals feel more urgent, more worthy of sustained attention, goals tied to achievement, to status, to proving capability. These felt essential, not optional. The prefrontal cortex allocated resources toward them automatically, because the dopamine signals associated with these goals were strong, clear, amplified by androgenic input. Now those signals are weaker. The prefrontal cortex still receives dopamine, still uses it to maintain focus, but the intensity has changed. What used to feel urgent now feels negotiable. What used to command attention now competes with everything else. The cortex hasn't lost its ability to focus. It's lost the chemical bias that made certain things feel automatically important. And without that bias, everything requires conscious effort, deliberate choice, active maintenance of attention rather than automatic capture. It's like trying to hold water in your hands. You can do it. But it takes constant attention, constant adjustment, and eventually some of it slips through. The prefrontal cortex filters information, decides what's relevant, what needs to be attended to, what can be ignored. Dopamine helps with this filtering. It marks certain inputs as salient, as worth processing, and testosterone used to influence which inputs got marked that way. Competitive challenges, physical tests, opportunities for dominance, these registered as important, as things that required immediate attention and planning. The prefrontal cortex prioritized them, built plans around them, allocated cognitive resources toward achieving them. Now, without testosterone's influence, these same inputs register differently. They are still noticed, still processed, but they don't trigger the same automatic prioritization. The prefrontal cortex treats them as one option among many, not as imperatives. And this changes how you move through the world. You notice opportunities, you consider them, you let them pass, not because you can't act, because acting doesn't feel necessary the way it used to. Working memory relies on sustained dopamine activity, the ability to hold information in mind while you use it, to remember what you're doing while you're doing it. The prefrontal cortex maintains this through constant low-level dopamine signaling, and testosterone supported that maintenance, made it easier to keep goals active, to resist distraction, to stay on task. Without that support, working memory becomes slightly less reliable. You walk into a room and forget why. You lose track of what you were saying mid-sentence. Not often, not dramatically, just occasionally, just enough to notice. The prefrontal cortex is still working, still maintaining representations, just with less dopamine support, less chemical reinforcement. And so the representations fade faster, become harder to sustain, require more conscious effort to keep active. Impulse control changes too. The ability to override immediate desires in favor of long-term goals. This has always required prefrontal activity, dopamine-mediated inhibition of subcortical impulses, and testosterone enhanced that inhibition for certain types of impulses. Aggressive ones, competitive ones, risk-seeking ones. These were easier to control when testosterone was higher, because the prefrontal cortex had the dopamine resources to maintain strong inhibitory signals. Now those resources are reduced. The impulses are still there, subcortical, automatic. But the prefrontal cortex has less capacity to override them consistently, not because it's damaged, because it's operating on a tighter dopamine budget, and so you might notice more impulsivity, or less, depending on which impulses the prefrontal cortex used to suppress, and whether those impulses themselves have also weakened with falling testosterone. The balance shifts, the cortex recalibrates. Some controls become easier, some harder. Nothing catastrophic, just different. Planning for the future requires holding distant goals in mind, imagining outcomes that haven't happened yet, making decisions based on possibilities rather than certainties. The prefrontal cortex does this through prospective memory, through maintaining representations of future states and using them to guide current behavior. And this process is dopamine dependent. Dopamine helps encode the value of future rewards, helps maintain motivation toward goals that won't pay off immediately. Testosterone amplified this for certain kinds of future rewards. Career advancement, competitive success, physical achievement. These felt worth working toward, worth sacrificing for. The prefrontal cortex could hold them in mind easily, consistently. Now the dopamine signals encoding their value are weaker. The future rewards still exist, still matter, but they feel more distant, less compelling. The prefrontal cortex struggles to maintain motivation toward them, not because it can't, because it requires more effort than it used to, more conscious intervention, more deliberate choice, and eventually some goals get abandoned, not consciously decided against, just quietly deprioritized, allowed to fade from the prefrontal cortex's active maintenance, released back into the space of things that might matter but don't feel urgent enough to pursue. The cortex is still capable, still functional, still able to plan, to focus, to resist distraction. It's just doing so with different chemical support. with less dopamine amplification, less testosterone modulation, and so its outputs change. What it decides is worth focusing on, what it maintains in working memory, what it prioritizes in planning. All of this shifts toward things that generate dopamine signals strong enough to register without androgenic amplification. Toward quieter goals, steadier ones that don't require the prefrontal cortex to work as hard to maintain them, ones that feel sustainable rather than urgent. And maybe that's acceptable. Maybe urgency was never sustainable anyway. Maybe the prefrontal cortex is just learning to work within its new limits, to allocate attention more carefully, to choose goals more selectively, to accept that not everything needs to be pursued, that some things can be let go without loss. The prefrontal cortex continues. It filters, it plans, it holds goals in mind. But it does so more selectively now, with less automatic prioritization, with fewer chemical signals telling it what must be attended to versus what can be released. The capacity remains, the function persists, just operating within a quieter dopamine environment, just learning to focus without the amplification that once made certain pursuits feel essential. The cortex adapts, it recalibrates. And in that recalibration, motivation becomes something you choose rather than something that chooses you. There's a moment happening right now inside a neuron in your Substantia nigra where a strand of DNA is being unwound by an enzyme, where the genetic code is being read and translated into messenger RNA, where that RNA will travel out of the nucleus and into the cytoplasm to serve as a template for building a protein. And this moment has happened countless times before in this same cell, this same gene being transcribed, this same protein being produced. But the frequency of these moments has been changing slowly, across months and years, because the signals telling the cell which genes to activate have themselves been changing. Testosterone used to enter this cell regularly. It would cross through the membrane, bind to an androgen receptor in the cytoplasm, and the testosterone receptor complex would move into the nucleus where DNA is stored. It would bind to specific sequences on the chromosome, androgen response elements, short stretches of genetic code that function as switches, and when testosterone bound to these switches, certain genes would turn on. Genes coding for tyrosine hydroxylase, the enzyme that makes dopamine, genes coding for vesicular monoamine transporter, the protein that packages dopamine into storage vesicles, genes coding for dopamine transporter, the protein that clears dopamine from the synapse after release. These genes would be transcribed more frequently, more messenger RNA would be produced, more protein would be built. The cell would have everything it needed to make dopamine efficiently, to release it quickly, to recycle it after use. But now testosterone arrives less often. The androgen receptors spend more time unoccupied. The androgen response elements on the DNA receive fewer activation signals. Gene transcription slows. It's not entirely clear how much this matters at first, whether the cell can compensate by keeping the existing proteins around longer, by recycling them more efficiently, by making each molecule of tyrosine hydroxylase work harder. But eventually the reduced transcription begins to show effects. The proteins start to decline in concentration, and the cell's capacity for dopamine synthesis decreases proportionally. The gene for tyrosine hydroxylase doesn't turn off completely. That would be catastrophic. The cell would stop making dopamine entirely. Instead, it's transcribed less often. The promoter region gets activated by fewer transcription factors. The rate of messenger RNA production drops by 10%, 20%, maybe more. And this reduced transcription rate means the cell maintains lower steady-state levels of the enzyme. Less tyrosine hydroxylase means less dopamine production per unit time. The cell still converts tyrosine to L-dopa to dopamine. It just does so more slowly. And when an action potential arrives, when the cell needs to release dopamine, there's less of it available. Fewer molecules packaged in vesicles, fewer vesicles ready to fuse with the membrane. The release happens, but it's smaller, quieter. There are other genes affected too. Genes involved in mitochondrial function and oxidative stress response and calcium handling, all of which contain androgen response elements, all of which were being transcribed at higher rates when testosterone was more abundant. And now, with reduced androgenic signaling, these genes are also being transcribed less frequently. Their protein products declining in concentration, and the cumulative effect is a cell that's less robust, less capable of handling metabolic stress, less efficient at managing the challenges of continuous neurotransmitter production. It's interesting how invisible this is. You can't feel gene transcription changing. Can't notice when a promoter region receives one less activation signal per day. But these microscopic changes in nuclear activity accumulate into macroscopic changes in behavior, in motivation, in how intensely you want things, and the path from one to the other runs through gene expression, through the process by which DNA instructions become cellular reality. The Substantia Nigra is involved primarily in motor control, in the smooth execution of movement, but it also contributes to motivation and reward processing. And the dopamine neurons here have androgen receptors just like the ones in the ventral tegmental area, and they respond to testosterone's decline in similar ways: reduced gene expression, reduced protein synthesis, reduced dopamine output. You might notice this as a slight heaviness in movement, a fractional delay before initiating action, not paralysis, not anything clinical, just a subtle change in how easily movement flows from intention, and this correlates with reduced dopamine signaling from the Substantia nigra, with neurons that are producing less neurotransmitter because the genes coding for dopamine synthesis machinery are being transcribed less often. Gene expression is regulated at multiple levels. Testosterone affects transcription, the first step, but there are also post-transcriptional controls, ways the cell can regulate how much protein gets made from a given amount of messenger RNA, and some of these controls are also influenced by androgens, meaning that falling testosterone can reduce protein production both by decreasing transcription and by making the transcription that does happen less efficient at producing functional protein. The net effect is multiplicative, compounding, and it's not entirely clear whether the cell tries to compensate, whether there are feedback mechanisms that sense reduced protein levels and attempt to upregulate transcription to maintain homeostasis. But if such mechanisms exist, they seem inadequate, because dopamine output does decline measurably when testosterone falls, does continue declining as long as testosterone remains low. The genes themselves haven't changed. The DNA sequences are the same as they've always been. The androgen response elements are still there, still functional. What's changed is how often they get activated, how often the transcriptional machinery gets recruited to read these genes. And this change in activation frequency, persistent across time, reshapes what the cell is capable of, not by altering its genetic potential, but by altering which parts of that potential get expressed at any given moment. The cell still contains all the instructions it needs to make dopamine efficiently. It just doesn't use those instructions as often anymore, doesn't activate the genes as frequently, doesn't produce the proteins in the quantities it used to, and so the cell's output changes, becomes less, becomes fewer, not because anything is broken, because the regulatory signals have shifted, because testosterone, which used to tell these genes to stay active, to keep producing protein, to maintain high-capacity dopamine synthesis, is no longer delivering that message with the same consistency it once did. The genes wait. They can still be activated, still be transcribed. They just need the right signal, the right transcription factor binding to the right promoter region. And when that signal is testosterone and testosterone is declining, the genes spend more time in their inactive state, more time waiting for activation that doesn't come, and the cell, reading the absence of activation as information, adjusts its operations downward to match the genetic instructions it's actually receiving rather than the ones it used to receive. Gene expression continues. Transcription happens, proteins are built, just less of them, less frequently. In response to a hormonal environment that no longer prioritizes dopamine production the way it once did, and the neuron, shaped by this new pattern of gene expression, becomes a quieter version of what it was, still functional, still producing dopamine, still participating in the circuits that generate motivation and movement, just doing so with reduced genetic support, reduced protein synthesis, reduced capacity. The DNA sits in the nucleus. The genes wait for activation. The androgen response elements remain ready. But testosterone arrives less often now, binds less frequently, activates transcription with longer gaps between signals, and in those gaps, in the silence where activation used to happen regularly, the genes produce less. The proteins decline. The cell adjusts its operations to match what the genetic instructions currently allow, not what they used to allow, what they allow now, in the absence of the hormone that once kept them active. 5 years ago, anticipation felt sharper. The hours before an important meeting, the moments before a competition, the space between wanting something and getting it. That anticipation lived here, in the nucleus accumbens, a structure no larger than a pea, tucked deep beneath the frontal cortex, part of the ventral striatum. Dopamine neurons from the VTA project here. They release neurotransmitter in response to predicted rewards, in response to cues that signal something good might be coming, and for years testosterone amplified those dopamine releases, made them sharper, stronger, more capable of capturing your attention completely. The nucleus accumbens learned to expect certain intensities, certain patterns of dopamine arrival. It built its response thresholds around those expectations, and now those expectations are being revised. The dopamine still arrives, the neurons still fire, but the peaks are lower, the surge is less pronounced, and the nucleus accumbens, reading these quieter signals, begins to recalibrate what it considers rewarding enough to pursue. The part that feels most fragile is the shift from wanting to liking. Wanting happens here, the drive toward reward, the motivation to pursue. Liking happens partly here too, but through different circuits, different neurochemical processes, and testosterone primarily influenced wanting. The anticipatory component, the surge that happens before you get something, not after. So what's changing most noticeably is how much you care about outcomes before they arrive, how much effort feels justified in pursuit of goals, how strongly the anticipation of reward pulls you forward. The nucleus accumbens operates through prediction errors. When something good happens that you didn't expect, dopamine surges. The nucleus accumbens learns to associate the cues that preceded that outcome with reward. When something good fails to happen when you expected it, dopamine dips. The nucleus accumbens learns that those cues were misleading, and over time this constant updating builds a model of the world, of what predicts reward, of what's worth pursuing. Testosterone used to make those dopamine surges larger for certain categories of reward, achievement, status, physical victory. These produce particularly strong prediction error signals. The nucleus accumbens learned they were extremely valuable, worth significant effort, but now the surges are smaller. The same achievements produce less dopamine, less teaching signal, and the nucleus accumbens, updating its model based on current feedback, begins to mark these outcomes as less valuable than it used to. Not worthless, just not as essential. The neurons here are densely packed with dopamine receptors, D1 and D2 receptors, both types concentrated in the medium spiny neurons that make up most of the nucleus accumbens. These receptors determine how the nucleus accumbens responds to dopamine, how it translates chemical signal into behavioral output. And as testosterone has declined, receptor density has changed. Some neurons have down-regulated D2 receptors, others have maintained them. The heterogeneity creates a system where some reward signals still register strongly while others barely register at all. The nucleus accumbens becomes selective in ways it wasn't before, more responsive to certain types of reward, less responsive to others. And what stands out most is the declining response to competitive rewards, to status-related outcomes, to the kinds of achievements that testosterone-amplified dopamine used to mark as supremely important. These still produce dopamine release, still activate the nucleus accumbens, just not enough to feel urgent, not enough to override other considerations. There's a sub-region called the core and another called the shell. The core is more involved in action selection, in choosing which behaviors to execute. The shell is more involved in emotional processing, in the affective component of reward. Both contain androgen receptors. Both respond to testosterone, and as testosterone has fallen, both have recalibrated, though perhaps not identically, perhaps with slightly different time courses or sensitivities, creating subtle shifts in how reward gets processed, in whether the emotional or the behavioral component dominates. You might find that things still feel pleasant when they happen, but you're less driven to make them happen. That's the shell maintaining function while the core becomes less responsive. Or you might find that you pursue goals efficiently once started, but starting feels harder than it used to. That's the core maintaining function while the shell provides less emotional pull. The nucleus accumbens doesn't work alone. It receives input from the prefrontal cortex, from the amygdala, from the hippocampus. It integrates information about goals, emotions, context, and it sends output to motor regions, to areas that translate motivation into action. Testosterone influenced this entire network, not just the nucleus accumbens itself. So as testosterone has declined, the whole system has shifted. The inputs arriving at the nucleus accumbens are different. The outputs it generates are different. The integration of motivation and emotion and memory produces different results than it used to. What remains constant is the structure, the anatomy, the pathways themselves. What changes is the chemical environment they operate in, the dopamine concentrations, the receptor densities, the gene expression patterns, and these chemical changes accumulate into functional changes, into a nucleus accumbens that expects less. That requires stronger signals to activate. That marks fewer outcomes as worth the effort required to achieve them. Years ago, this region would light up in anticipation of almost any competitive challenge. The dopamine would surge, the wanting would intensify. You'd feel pulled toward the challenge, compelled to engage. Now the same challenge produces a smaller surge, a quieter wanting. You notice it, consider it. Often choose not to engage, not because you've lost capability, because the nucleus accumbens is no longer marking it as sufficiently rewarding. The prediction error signal is too small, the anticipated dopamine release too modest. And without that chemical pull, without the nucleus accumbens insisting this matters, the challenge becomes optional. Something you could do, not something you must do. The graph of your internal life has flattened. The peaks are lower, the valleys less deep. Experiences that used to spike dopamine activity now produce gentler increases. Disappointments that used to crash dopamine signaling now produce modest decreases. Everything operates closer to baseline, more stable, less volatile. And the Nucleus accumbens, reading this new pattern, adjusts what it considers normal, what it expects, what it uses as reference when evaluating whether new experiences count as rewarding or disappointing. The baseline has shifted downward, the range has narrowed, and within that narrower range, life continues. Rewards still register, motivation still exists, just quieter, just less insistent, just enough to keep functioning without the sharp edges that testosterone-amplified dopamine used to provide. The Nucleus accumbens waits for dopamine, receives it, processes it, translates chemical signal into the felt sense of wanting something. And that translation, once immediate and intense, has become more measured, more restrained, more conditional on whether the dopamine signal actually reaches threshold, rather than assuming everything worth wanting will. The Nucleus accumbens continues its work, translating dopamine into desire, processing reward prediction, updating its model of what matters, but the model has changed. The predictions have adjusted, the thresholds have risen, and what it no longer expects, what it no longer marks as essential, quietly fades from the category of things worth pursuing. Not lost, just reclassified, just moved from urgent to optional, from compelling to merely possible. The wanting remains, just gentler now, just operating within the reality of smaller surges, lower peaks, a graph that's learned to flatten itself around what dopamine can actually provide when testosterone is no longer amplifying the signal. The brain is solving a problem. Dopamine signaling has weakened. Testosterone no longer amplifies it. Motivation needs to come from somewhere, so other systems begin to contribute. Quietly, without announcement. Serotonin pathways activate more readily. Neurons in the raphe nuclei deep in the brainstem increase their firing rate slightly. They release more serotonin into cortical regions, into the hippocampus, into areas that regulate mood and contentment. Serotonin doesn't create the same urgency dopamine does. It doesn't generate sharp wanting, doesn't produce the feeling that something must be pursued immediately. Instead, it offers steadiness, a sense that things are acceptable as they are. This is compensation, though not replacement. The brain is reorganizing around what's available, and what's available is serotonin, more of it than before, filling some of the space dopamine used to occupy. You might notice this as increased acceptance, less agitation about outcomes, less restlessness when goals aren't met. A quieter form of satisfaction that doesn't depend on achievement. The serotonin system was always there, always functional. It just wasn't as prominent when dopamine was dominating motivational circuits. Now, with dopamine quieter, serotonin's influence becomes more noticeable, more central to how you experience the day. Norepinephrine plays a role too. Released from the locus coeruleus, another brainstem nucleus, norepinephrine regulates arousal, attention, response to stress. When testosterone was higher, dopamine handled much of the motivational arousal, the internal sense that something needed attention. That action was required. Now norepinephrine compensates partially. It increases baseline arousal levels, makes the nervous system slightly more responsive to environmental demands. This doesn't feel the same as dopamine-driven motivation. It feels more like vigilance, like being ready to respond if needed, less about pursuit, more about presence. The endogenous opioid system activates differently now. Beta-endorphin, enkephalins, dynorphins, These internal opioids regulate pleasure, pain, social bonding. They were always part of reward processing, but testosterone used to overshadow them in certain contexts, in competitive situations, in achievement-oriented scenarios. Dopamine surges were so strong that opioid contributions barely registered. Now the opioid system has more influence. Physical contact feels more rewarding. Social connection generates stronger positive feelings. The pleasure derived from these experiences doesn't depend on dopamine intensity. It depends on opioid receptor activation, and those receptors are still functional, still capable of producing satisfaction, just a different kind, softer, more relational than goal-oriented. Oxytocin pathways strengthen too. Released during social bonding, during moments of trust and connection, oxytocin enhances the reward value of interpersonal experiences. When dopamine was amplified by testosterone, competitive achievement often felt more rewarding than social connection. The dopamine system prioritized individual success over collective belonging. Now, with dopamine signaling reduced, oxytocin-mediated rewards become more salient. Time spent with people you trust feels better than it used to, more satisfying, more central to well-being. This isn't cognitive reframing, it's neurochemical rebalancing. The brain is shifting which systems generate the majority of positive affect, moving from dopamine dominant to a more distributed model, where multiple neuromodulators contribute to the experience of satisfaction. GABA activity changes in subtle ways. The brain's primary inhibitory neurotransmitter, GABA, reduces neural excitability, promotes calm. With less dopamine-driven urgency, with less constant push toward goal pursuit, GABA's influence can be felt more clearly. A sense of ease that doesn't require justification, that doesn't need to be earned. Just the baseline state when excitatory systems aren't constantly activated. These compensatory systems don't restore what testosterone-enhanced dopamine provided. They don't recreate urgency, don't generate the sharp-edged drive that characterized earlier decades. But they do provide something, a different form of motivation, one based on steadiness rather than intensity, on connection rather than achievement, on presence rather than pursuit. The emotional texture shifts. Life feels less like a series of peaks to chase and valleys to avoid, more like a plateau, stable, manageable, not exciting in the way dopamine surges were exciting, but not empty either, just differently satisfying. The brain has reorganized its reward architecture, distributed the work of generating positive affect across multiple systems, and in doing so created a new baseline, one that doesn't depend on testosterone, doesn't require dopamine intensity, one that operates through serotonin's contentment, norepinephrine's alertness, opioids' warmth, oxytocin's connection, GABA's ease, these systems working together, not perfectly replacing what was lost, but providing enough to keep functioning, enough to find meaning, enough to experience satisfaction even when it doesn't surge. The compensation is incomplete. Some aspects of testosterone-enhanced dopamine function simply can't be replicated by other systems. The competitive drive, the physical urgency, the need to dominate, these fade without full replacement. But other aspects, the capacity for pleasure, for meaning, for connection, these remain, just sourced differently, generated through different pathways, felt through different neurochemical lenses. And perhaps this is what aging does at the neurochemical level. It shifts the balance, reduces reliance on any single system, distributes function across multiple networks, creates redundancy, resilience, so that when one system weakens, others can contribute more. The brain doesn't fail, it adapts. It finds new ways to generate the experiences that make life worth living. New pathways to satisfaction, new sources of motivation, quieter ones, steadier ones, but still sufficient, still capable of sustaining you through the days, through the gradual recalibration, through the slow, ongoing process of becoming someone who wants things differently than before. Not less, just differently, through systems that were always there, just waiting for their turn to matter. The compensatory pathways activate, serotonin steadies, norepinephrine attends, opioids warm, oxytocin connects, GABA calms, and together these systems create a new baseline, one that doesn't surge, one that doesn't demand, one that simply provides enough to continue. Not the same as what dopamine used to offer, but sufficient, present, available. The brain solves its problem, not by restoring what was, but by reorganizing around what remains. And in that reorganization, life continues, different, quieter, but still capable of satisfaction, still capable of meaning, just through pathways that don't require urgency to function. You're still here, after everything that's changed at the cellular level, after all the receptor adjustments and gene expression shifts and synaptic remodeling, after dopamine has quieted and testosterone has declined and the compensatory systems have reorganized themselves. You're still here. The neurons that hold your memories are intact. The patterns that constitute your identity haven't dissolved. What's changed is the chemical context those patterns exist within. The background state, the baseline mood, the ease with which certain thoughts and behaviors activate. But the fundamental structure remains. You've probably noticed by now that some things feel harder. Starting new projects, maintaining competitive drive, summoning urgency when nothing feels urgent. These changes are real. They're neurochemical. They're happening in dopamine pathways that have recalibrated around falling testosterone. But other things remain surprisingly stable. Your ability to learn, to reason, to connect information across contexts, these don't depend primarily on dopamine. They depend on broader cortical networks that testosterone doesn't directly modulate. So even as motivation changes, cognition persists. Even as drive weakens, understanding deepens. The shift isn't toward incapacity, it's toward a different mode of being, one that doesn't rely on intensity to function. Your personality hasn't changed at its core. The traits that defined you, curiosity, caution, openness, conscientiousness, these emerge from networks far more distributed than dopamine circuits alone. What's changed is how those traits express themselves. Curiosity becomes less restless, more patient. Conscientiousness becomes less perfectionistic, more accepting of good enough. The underlying structure remains, the surface expression adapts. Relationships often deepen during this transition. When competitive drive fades, when the need to prove yourself weakens, space opens for different kinds of connection. Less about status, more about understanding, less about what people can provide, more about who they are. The oxytocin and opioid systems that now contribute more to reward processing, they favor these deeper connections. They generate satisfaction from presence rather than achievement, from being with rather than doing for. And many people find this shift quietly profound, a relief they didn't know they needed. Meaning doesn't disappear when dopamine quiets. Meaning emerges from narrative, from the stories you tell about your life, from the connections you draw between experiences. These are cortical processes, hippocampal processes, prefrontal processes. They continue regardless of dopamine baseline. What changes is which experiences feel meaningful enough to incorporate into those narratives. Achievements matter less. Relationships matter more. External validation matters less. Internal coherence matters more. The brain is reorganizing around what remains valuable when urgency isn't available to mark things as important. And what remains is often simpler, more fundamental: connection, understanding, presence. The capacity for joy still exists. It just activates through different pathways now. Less through dopamine surges in response to achievement, more through endogenous opioid release during moments of warmth, through serotonin-mediated contentment with the present, through the quiet satisfaction of having enough rather than needing more. These aren't lesser joys, just different ones, more sustainable, less dependent on external circumstances, available even when nothing dramatic is happening. Physical sensation changes too, though subtly. Movement feels different when dopamine signaling from the Substantia nigra has decreased. Less automatic, requiring slightly more conscious initiation. But this increased awareness can become a gift. You notice your body more, the small adjustments, the balance. Presence in physical experience deepens even as physical urgency fades. What you're experiencing isn't decline in any totalizing sense. It's reorganization, recalibration, the brain adapting to a new hormonal reality. And in that adaptation, some capacities weaken, while others strengthen. Some experiences become harder to access, while others become more available. The net result isn't uniformly positive or negative. It's complex, mixed, deeply individual, but beneath the complexity, beneath the mixed results, something fundamental persists: the capacity for experience itself, for awareness, for choosing how to respond, for finding meaning in whatever circumstances exist. This doesn't depend on dopamine levels or testosterone concentrations. It depends on consciousness, on the irreducible fact of being present, and that hasn't changed. The neurochemical weather has shifted, the background state has recalibrated, but the capacity to be here, to notice, to respond, to care, that continues, not louder, not quieter, just continuing, as it always has, as it always will. Beneath every surge and every silence, beneath urgency and ease, beneath the dopamine and the testosterone and all the compensatory systems working to maintain equilibrium, you remain, changed in how you experience, unchanged in that you experience. And perhaps that's enough. Perhaps it's always been enough. The rest is just chemistry, important chemistry, chemistry that shapes everything. But beneath the chemistry, something steadier persists. Something that doesn't need to be urgent to matter. Something that continues even as everything else recalibrates. You, still here, still capable of meaning, still present. The dopamine has quieted. The testosterone has declined. The systems have compensated. And through it all, through every cellular adjustment and every synaptic remodeling, you've remained. Not unchanged, but undiminished, still capable of experience and still capable of meaning, still here. And that continuity, that persistence beneath all the neurochemical flux, is perhaps the most fundamental truth of all. The rest is just biology, important biology, but still just biology, and you are more than the sum of your neurotransmitters. You always have been. Thank you for spending this time here, in the quiet space where biology unfolds. If you found something valuable in these hours, if this exploration brought you a different kind of understanding, consider supporting the Sleepy Biologist through channel membership or super thanks. Your support allows these long, unhurried journeys through the body to continue. And if you'd like to share where you're listening tonight or what brought you here, the comments are always open. I read them, I appreciate them. Until next time, rest well.